If a live or viable seed has the right environmental conditions, it can germinate. But in order for it to germinate, there are a series of events that have to happen in sequence so that the seed can produce a seedling. It hopefully develops into an adult plant capable of photosynthesizing and reproducing and making an additional generation. So let's talk about what these steps are. First of all, this is straight out of your handouts. The plant has to uptake water, or in this case, the seed has to uptake water, because as we said before, seeds commonly are in at least a semi-dehydrated state because that slows down respiration and that increases the so-called shelf life of the seed. And so seeds tend to be dehydrated, and so they have to hydrate. They have to do this so that the testa softens. This is particularly important for plants that have a hard, thick outer seed coat. And the protoplasm hydrates. Protoplasm just means the living cells hydrate. In other words, their cytoplasm increases so that solutes can be moved around and all the biochemical processes can start happening. As it says in the notes, this process is largely physical, and that just means that it's going to occur via osmosis. Water, as you recall, moves from areas of high concentration to areas of low concentration. And so water moves from the relative high concentration in the soil to the relatively low concentration region of the interior of a seed. And this will occur whether or not the seed in question is actually even alive or not. Even dead seeds will uptake water via this osmosis process. And for those with hard outer seed coat, a lot of times frequent wetting and drying cycles might be necessary to actually break the outer testa to the point where the seed can expand and uptake its full needs in terms of water. Once the cytoplasm of the cells has been rehydrated, they can start cellular activity. And what they do, as outlined in the notes, is they start to produce specific enzymes that are going to be needed for the next step, and respiration increases. So respiration goes up, enzyme production begins, and this is, as you may recall when we talked about the endogenous hormones, gibberellic acid is thought to be heavily involved in this process. So this stimulates that production of enzymes. So we'll say GA stimulated, GA being gibberellic acid. Well, what does it need all these enzymes for? Well, it needs it for digestion. The enzymes are used to break carbohydrates down into a more usable form. Typically, we're talking about breaking down starches. So enzymes break starches, S, down into sugars. Starches aren't particularly soluble in water, and by breaking these starches down into sugars, which are soluble in water, they can be transported to the growing parts of the seed, or now we would call a seedling, once this process gets started. And so all of this above adds up to growth, and we get rapid cell division, elongation, and eventually differentiation into different tissues. And finally, we get a root and shoot radical emerging from the seed. And we can see that this is what has started to happen in this seed as it germinated. This is a pinto bean. We had a seed coat here, or testa, that softened once this seed was exposed to moisture. And over the period of a couple of days, it hydrated to the point that the cytoplasm hydrates, enzymes start being produced to break down the starches in these cotyledons, which are the main storage tissue for most beans. You can see the first true leaves and the primary root that has begun to grow. And what would happen as this process continues is the roots would branch out, okay, and anchor this into the soil and begin to push the shoot, or this hypocotyl, which is what we call the initial shoot tissue here, or the original stem of the plant, to push these cotyledons above the soil surface. Let's say that's the surface. so that these first true leaves can be exposed to sunlight. And these are the ones that are going to start conducting photosynthesis. The cotyledons are the first leaves. They're the seed leaves, once again, but they typically are not capable of photosynthesizing. These smaller leaves inside that are going to emerge last are what are going to conduct photosynthesis for this developing seedling. So, summarize once again. The seed in its dry state has to hydrate, and that is taken care of by osmosis. If there is moisture in the soil, it tends to move right into the seed. Next step would be to start cellular activity, specifically the production of enzymes, and this is stimulated by gibberellic acid. These enzymes break down complex carbohydrates such as starches into sugars. In some cases, they're also breaking down things like fatty acids and oils, also used by plants to store energy long term, to produce sugars, or at least get a usable fuel for respiration out of those lipids. And then finally, growth starts occurring. We have the growth of a root and a shoot, and if all goes well and continues to stay that way, this seedling can become a mature plant, capable itself of producing more seeds.